Hello everyone, I'm Lowens A. Lapitan of Group Stripes. Today, I am going to discuss or introduce the bacteria Serratia. The general characteristics of Serratia species, it belongs to the Enterobacteriaceae family, a gram-negative bacilli. It is non-spore forming and facultatively anaerobe. This bacteria is motile with the help of its flagella. Some are capsulated, and some species produce pink or red pigments. As you can see, Serratia species are an opportunistic pathogens because it is associated with outbreaks in healthcare settings. Most Serratia species are non-pathogenic, but those pathogenic ones can cause infection to those immunocompromised people or those people whose immune system is weak. The main pathogenic species of Serratia species is the Serratia marcescens. It infects animals, plants, and humans. Serratia species ferment lactose slowly and positive for ONPG tests. They can be differentiated from other tribes by their ability to produce extracellular DNase. And these species also produces enzymes just like lipase and gelatinase. Serratia species had 8 significant species. These are Serratia marcescens, Serratia liquefacens, Serratia rubidiae, Serratia odorifera, Serratia plymoutica, Serratia ficaria, Serratia entomophila, and Serratia fonticola. Serratia species had virulence factors. These are proteoglycan, biosurfactants, DNase, lipase, protease, gelatinase, hemolysin, chitinase, chloroperoxidase, and alkaline phosphatase. Proteoglycan is a growth pigment used as a phenotypic identification marker for Serratia species due to its red colorization. Biosurfactants are sourced for antimicrobial, antifouling, and anti-tumor compounds that possess emulsification and surface activity. The DNase allows the bacteria to exploit the environment and maximize the availability of nutrients. The lipase, protease, and gelatinase cause contact lens-related corneal ulcers in humans. Chitinase is used as defense mechanism against plant pathogen. The chloroperoxidase allows hydrolysis of phosphodiester bonds. The alkaline phosphatase involved in cell signaling processes. The selective medium for the isolation of Serratia species is the Caprylitalus Ct agar. That is why this medium is used because Serratia species use caprylic acid as their carbon source. Serological typing and different types of polymerase chain reaction can also be used in identifying the Serratia species. We can also use biotyping, phage typing, and ribotyping. For differentiating non-pigmented Serratia strains from other Enterobacteriaceae, we use the selective medium called Twin80 medium. Serratia hydrolyzes the Twin80 medium with the enzyme esterase, resulting to releasing of free fatty acid. For differentiation and isolation of Serratia species, we can also use DNase agar, which contains DNA, and CTC agar, which contains cephalotin. Cephalotin is some Serratia species are resistant to it. On blood agar and other media, S. marcescens produces red colonies. On CT agar, Serratia species produces small and slightly bluish white. On Twin80 medium, Serratia species produces large, pinkish, and with a white zone of precipitate. On this part, I am going to introduce the 8 significant species of Serratia. The first one would be the Serratia marcescens, which is the main pathogenic species of all. Serratia marcescens is commonly found on soil, water, plants, and animals. It is widely found on non-potable water of underdeveloped countries due to poor chlorination. Also, Serratia marcescens can be found in toilet, showers, and wetted tiles. These type of bacteria cause infection in immunocompromised people, and this bacteria is known to grow on disinfectant, antiseptic, and distilled water. Serratia marcescens is motile due to its flagella. It is a rod-shaped and a gram-negative bacillum and facultatively anaerobe. This bacteria produces red pigmentation and have biofilm. Serratia marcescens is involved in hospital-acquired infections, particularly catheter bacteremia. It is commonly found in the respiratory and urinary tract of hospitalized adults and the gastrointestinal of children. The urinary tract is the most common site of infection for S. marcescens, and it can be spread due to the hands of the personnel inside the hospital. These are the disease association if a person is infected with Serratia marcescens: sepsis, bacteremia, cerebral abscess, osteomyelitis, UTI, and ocular infections. And these are the common diseases of Serratia marcescens. The first one is meningitis, which is the inflammation of the membrane surrounding the brain and the spinal cord. The second one is the endocarditis, which is the inflammation of heart muscle. And the last one is the pyelonephritis, which is the inflammation of kidney. We can diagnose S. marcescens by taking biopsies of infected tissue, mucus, or blood. The next bacteria of Serratia species is the Serratia liquefaciens. It is a gram-negative rod-shaped bacterium which is motile due to its Petricus flagella. It is facultatively anaerobe. It is commonly found on plant species, more specifically in the rhizosphere. Serratia liquefaciens is a widespread bacterium incapable of colonizing plants, soil, and water, and digestive tract of rodents, insects, fish, and humans. The diseases that are associated with this bacteria are mastitis in dairy herds and nosocomial infection. The next bacteria of the Serratia species is Serratia rubidae. It is a gram-negative rod-shaped bacterium which is motile due to its Petricus flagella. Serratia rubidae is a potential strain for lipase production. This bacteria is noted for producing a characteristic of pink to red pigmentation when cultured in a room temperature. When a person is infected with S. rubidae, that person might acquire urinary and respiratory tract infection. The next bacteria of the Serratia species is the Serratia odorifera. It contains two biogroups, which are the Serratia odorifera biogroup 1 and Serratia odorifera biogroup 2. The Serratia odorifera emits dirty and musty odor that resembles a rotten potato. Serratia odorifera biogroup 1 is a gram-negative rod-shaped bacterium which is motile due to its Petricus flagella. It is isolated in the respiratory tract, and it is distinguished with other group by its fermentation of sucrose and raffinose and with the appearance of ornithine decarboxylase. Serratia odorifera biogroup 2 is a gram-negative and rod-shaped bacterium which is motile due to its Petricus flagella. It is isolated in the blood and the CSF. It can be distinguished with the other groups because it does not ferment sucrose and raffinose, and it doesn't have the ornithine decarboxylase. The first case in which the organism is found to be invasive in humans was when it was isolated in the urine sample of an alcoholic male with cirrhosis and septic shock. The next bacteria of the Serratia species would be the Serratia plymutica. It is a gram-negative, catalase-positive, oxidase-negative, and a non-motile bacterium. Serratia plymutica was isolated from a biofilm that formed on a pasteurizer plate of a sanitized milk processing line. This species of Serratia is used as a controlled biological agent for fungal plant pathogen in agriculture. The habitat of S. plymutica is predominantly water, but it has been isolated on soil, water, insect, plants, and sometimes in the clinical samples. This type of gram-negative bacterium is associated with plants. It is isolated on the rhizosphere of wheat, oat, cucumber, and potato. The disease that is associated with Serratia plymutica is nosocomial septicemia. The next bacteria in the Serratia species would be the Serratia ficaria. It is an enterobacterium that involved in the fig tree ecosystem. The Serratia ficaria is isolated in the rare occasion of infection in the digestive system, including the gallbladder and the bile duct. This kind of Serratia species is associated with the fig wasp biological cycle. The diseases that are associated with Serratia ficaria would be urinary and respiratory tract infections. The next bacteria of the Serratia species is Serratia entomophila. It is gram-negative, non-spore-forming, non-capsulated, and a rod-shaped bacterium with a petricus flagella. It is an insect pathogen, and this is a species that uses itaconate. The bacterium Serratia entomophila is a natural occurring pathogen. It caused the amber disease of the New Zealand grass grub, and there is no strain that has been identified to be involved in human, animal, or plant diseases. The last bacteria in the Serratia species would be the Serratia fonticola. It is a gram-negative, motile, facultatively anaerobe bacterium and rod-shaped. It expresses phosphate solubilization and antifungal activity against Rhizoctonia species. Serratia fonticola can be isolated on the soil and water, and it can be isolated on the rhizosphere of pea roots. This type of bacteria cause illness on person with weak and damaged immune system. Also, it can cause skin and tissue infection following with a trauma. And that concludes our discussion about the Serratia species. Thank you.